Hi everyone. In this video, I'll be talking about our work on connecting cell and tissue phenotype to protein expressions using canonical correlation analysis or CCA. I'm Vaishnavi and this is joint work with Ben from CMU and my PhD advisor Mindo from UIUC. With the recent advancements in technology, we are now able to obtain spatially resolved information about gene expression and protein expression. In this work, we focus on spatially resolved protein expressions or spatial proteomics data. Spatial proteomics data captures the protein expression coming from a cell, and we are able to locate not only which cell the protein is being expressed in, but also where in the cell is the expression arising. Given this spatial proteomics data, we can extract two different types of information at the cell level. One, the nice thing to do would be to aggregate information about the protein expression across the entire cell, which results in a protein expression level for a given cell. Correspondingly, spatial proteomics data often results also in images which allow us to capture morphological properties of a cell, such as the area and other shape properties, the radial distribution of proteins within a cell, which capture where in the cell is a given protein being expressed, as well as the relation between different cells and tissue quantified through neighborhood properties. Given this cell and tissue phenotype, as well as the protein expression levels arising from the same tissue corresponding to the same patient, we would like to understand what is the relation between these two different feature sets. To understand this relation, we make use of a correlation tool called CCA or canonical correlation analysis, which computes linear correlations between two given feature sets. Additionally, the CCA tool also results in CCA-based embeddings, which can be used for downstream analysis. The CCA-based embeddings can be obtained for each cell in the data and can be visualized in an underlying dimensionality reduced space, for example, using UMAP. This embedding space can be visualized with ground truth overlays of cell type or cell subtype information. Furthermore, this embedding space can also be clustered to obtain CCA clusters, which can then be overlaid on tissue in an in situ visualization of CCA clusters, or be used to quantify the composition of a given tissue corresponding to different CCA clusters. What that means is, given a tissue, we know each cell, which CCA cluster each cell belongs to, and we can use this information to obtain a tissue level characterization of the different CCA clusters. We apply a proposed method on spatially resolved proteomics data arising from multiplexed ion beam images or MIBI images from DCIS, a type of breast cancer. Below, you see examples of spatially resolved protein expression overlaid on tissue, as well as the corresponding cell segmentations provided in the data, and an overall view of the data shown on the bottom right. Our proposed method can be applied across all the cells available in the data, and the corresponding CCA weights can be visualized to understand feature importances corresponding to proteins as well as the phenotype information. The embedding space resulting from CCA can be visualized using ground truth cell type information as shown on the bottom right, or using the CCA clusters as shown on the bottom left. The composition of tissue based on the CCA clusters can be visualized in these composition plots, which allow us to compare the tissue originating from different humans. Here, the compositions are grouped by non-progressor cancer patients, progressor cancer patients, as well as normal humans. To understand what differentiates a seemingly similar cluster, a seemingly similar sample from a non-progressor versus progressor stage, we can compare the corresponding spatial overlay in situ plots and understand the spatial differences between the two different settings. A similar analysis can also be performed by focusing on only epithelial tumor cells available in the data. Here, we also look at the radial distribution of features as we discussed earlier, and the corresponding importances of different features are shown in the plot below. The corresponding CC embedding space can be visualized through the U-maps as shown below, and we notice here a concentration of different cells within a given subtype of a tumor. A further analysis reveals that these lumps are actually subclusters within a tumor subtype available as ground truths in the data. As a summary, we propose CCA as a robust analysis tool to discover connections between single-cell proteomics phenotype and protein expression data. We saw how CCA, when applied to all cells or just focused on epithelial tumor cells, allows us interesting analysis. And this is an indication that with the growth of newer technology and newer data, we need more exploratory studies which look at connections between phenotype and functional properties, and also methods which focus on how we can extract diverse information from this rich data to understand diseases such as cancer and other complex diseases. Thank you for watching.